Hello my dear friends, welcome to Live Prep. In the previous lecture, we have studied about there are two types of nucleic acids, they are DNA and RNA. And after that we studied about what are the various roles of DNA and RNA. Yes or no? So let us quickly revise that point and then finally we will come back to next remaining content. Okay. So let us quickly go through that what we have studied in the previous lecture. So in the previous lecture we studied that DNA has only one function. So wherever DNA is there, always DNA acts as genetic material. And here we studied about examples also. What are the organisms in which DNA acts as genetic material? So here as per NCERT what we studied, most of the organisms we studied, yes or no? And what this most includes? It includes all the five kingdoms. And along with this there are some viruses, not all viruses. Some viruses have DNA as genetic material. This is the point we have studied for DNA. And now let us quickly go for RNA. So in the case of RNA, so what are the various functions or roles of RNA? RNA acts as a genetic material. RNA can also act as genetic material, not only DNA. RNA can also act as genetic material. But in what type of organisms RNA will act as genetic material? This is important. Already all the five kingdoms, that means all living organisms, that means all unicellular and multicellular, that means all prokaryotes and eukaryotes have DNA as genetic material. Yes or no? Then what are remaining? Viruses. Viruses are not present under any of the kingdoms. And even in the viruses also, some viruses have DNA as genetic material, whereas some other viruses will have RNA as genetic material. So here we are writing again some viruses. Please remember here. So you should be little bit more careful about the word some. If they are giving the word all viruses, it will be wrong. Please remember this. So some viruses have DNA genetic material, some viruses have RNA as genetic material. And here we studied some examples. So TMV, tobacco mosaic virus, and then QB bacteriophage, and also HIV, human immunodeficiency virus. So these are the some examples that we have studied. And next one is RNA is acting as messenger. RNA is also acting as messenger. And what is the example for this? mRNA, messenger RNA. It is having information for protein synthesis. Yes or no? Based on the DNA, mRNA is produced and it is carrying some information that is needed for protein synthesis. Slowly in the next coming lectures we will study this. Not only this, RNA is also acting as adapter. What is the meaning of adapter? Adapter means connector. It is connecting two things. What are the two things? One side it will connect amino acid, other side it will connect codons present on mRNA. So since it is connecting two things, it is called as adapter. And who will act as adapter? tRNA. tRNA, what this T indicates? Transfer RNA. It is transferring amino acids needed for protein synthesis. That's why it is called a transfer RNA, adapter RNA. There is another name that is called soluble RNA that we will study in the regular chapter. Okay, regular lecture. Next one. It is also having some other role. What is this role? Sometimes some types of RNA are acting as catalysts. Catalytic role. Catalytic role means what? It is acting as an enzyme. Generally, what are the ones that are acting as enzymes? Generally, proteins are acting as enzymes. But here it is exception. RNA is acting as an enzyme. So, if RNA acts as an enzyme, it is called as ribozyme. If RNA acts as an enzyme, it is called as ribozyme. And for this, we wrote three examples. What are those three examples? 28S rRNA present in 60S subunit of 80S ribosome. This is one example. Second example is 23S rRNA present in 50S subunit of 70S ribosome. That is the second example we studied. And the last example we studied is snRNA. And what is its role? It is involving in splicing. Anyhow, what is this peptidyl transferase activity? These two are having peptidyl transferase activity. And what is peptidyl transferase activity? And what is the role of that one? All those things we will study under translation process. And this is splicing snRNA, this we will study under transcription process. So simply remember the examples of this, and with this part is completed. Yes or no? And not only this, now messenger, adapter is over. Now one more is there that we will see in the next one. And it is also having some structural role. It is also having some structural role. And what are the structures formed? Ribosomes. So in the ribosomes, RNA is present. The RNA that is required for formation of ribosomes is called as ribosomal RNA, rRNA. So these are some of the important functions of RNA that we are studying here. So with this, the role of RNA are completed. Clear? And after this, now we will go with NCERT content. In the NCERT, what are the other points they are saying? Nucleic acids, whether it is DNA, RNA, whatever it is, they are the polymers of what each unit is called as? Each unit is called as a nucleotide. So that's why we are writing that these are polymers of nucleotides. These are the polymers of nucleotides. Very, very important. Already in the first lecture itself, I told you this. Yes or no? When we have revised that biomolecules content, so that time we have studied this. Nucleic acids are polymers of nucleotides. Proteins are polymers of amino acids. Polysaccharides are polymers of monosaccharides. Already we have studied this. Same thing. Okay. This is one thing. And the second thing they are saying that, so in this case, so if you want to study the nucleic acid structure, first you need to study about the nucleotide structure. First you need to understand what is the structure of one nucleotide. If you know one nucleotide, then all the nucleotides are almost in the same way. Now once we know about each nucleotide, once we are start combining these two, then you can understand a long molecule. So first you need to understand the structure of one nucleotide. So you will know structure of all the individual nucleotides, and then you need to connect this. That's it. So once you are connecting all these things, a long polymer called nucleic acid is produced. If they are related to DNA, so then the molecule will be DNA. If these nucleotides are related to RNA, now the molecule formed will be RNA. That's it. So that is the reason why now what we need to do, if you want to study the DNA or RNA, first you need to study individual unit called nucleotide. So first we need to study about the nucleotide. But before directly going with that, in the NCERT they have given some other few points. Let us quickly go through these things, and then we will read NCERT content, and then we will go with nucleotide. In this lecture itself we will start with nucleotide. So here in the NCERT what they are saying, since it is the introduction, they are saying that in this chapter we will study all these things. This is the big list they have given. So this is a very, very big chapter, and this is the biggest chapter in entire biology of class 11th and class 12th. Please remember this. So that's why it will take so much of time, so many important concepts are there. So that's why we should be a little bit more careful, because its weightage in the examination is also very huge. Yes or no? So that's why let us go through this. So in this chapter we will study about structure of DNA. So anyhow, first we need to study structure of nucleotides, and after combining nucleotide we will get DNA structure. Yes or no? And here there is a model that is called Watson and Crick double helical DNA model is there. Already you know about this. Even in class 10th also you have studied that. So once again we will study this, and after that we will study about DNA packaging. So DNA is a very long molecule. How this much of long molecule is present inside a small cell? So that particular process is called as a packaging. That also we will study in this particular chapter. Next one, DNA replication. What is this DNA replication, transcription, translation? These three terms. In simple, I will try to explain you what is this. What is DNA replication? What is the meaning of replica? Replica. What is the meaning of replica? Replica means one more copy. So if there is a DNA and DNA is making one more copy of DNA, that means initially, suppose it is the double stranded DNA, one copy is there. From this one copy, finally what happens? Two copies will come. So this is the one, this is the one. So in this way, initially there is only one copy. From this one more is produced. In this way, ultimately two copies are produced. So in this way, making one more copy of DNA is called as replication, replica formation. That's why it is called as replication. So from the DNA is produced. It is called as DNA replication. This process is called as a DNA replication, and you can also use the word called DNA duplication. Duplicate means what? Same meaning, one more copy. So making one more copy of DNA is called DNA duplication or DNA replication. DNA duplication or DNA replication. Any word you can use. Now, after this, what is transcription? From the DNA, RNA is produced. It may be mRNA, tRNA, rRNA, snRNA at least. Now you know the name of snRNA also. Any type of RNA. Synthesis of any type of RNA from the DNA is called as a transcription. This process is called a transcription. And then if it is mRNA, messenger RNA, it has information for protein synthesis. So that now this is most important for protein synthesis. Anyhow, please remember, not only mRNA, tRNA, rRNA are also having different other roles in protein synthesis. Those anyhow in the regular chapter we will study. Now let us see this. The process of synthesis of protein from mRNA is called as a translation. Translation. That's why in simple, protein synthesis means what? Translation process. Protein synthesis means translation process. RNA synthesis means transcription process. DNA from DNA means DNA replication process. So all these things we will study. This is what they are saying here. And not only this. So how this protein is produced from mRNA, and here there is a concept called as genetic code. Genetic code. Actually, this genetic code is very important. In the protein, many amino acids are present. Suppose this is one amino acid, second amino acid. Protein is a polymer of amino acid. That means amino acid should be attached to each other. Now there is one more amino acid. Now there is one more amino acid. So here, so what type of amino acid will come here? Actually, that is decided by codon that is present in mRNA. So that's why genetic code concept also will come. Don't worry, this is just introduction. Simply you know that we will study about DNA replication, transcription, translation, genetic code. That's it. Don't worry about how exactly it is working. That's what actually we are studying in this chapter. This entire chapter is mainly about these processes only. Yes or no? Around 70% of the time will take for this process only, because they are very, very huge, highly conceptual. So that's why slowly we will study and understand all the things. This is just an introduction. Here they are saying that we will study DNA replication, we will study transcription, we will study translation, we will study genetic code and the role of genetic code in the determination of amino acid sequence based on mRNA. That also we will study. And after understanding all these things, how the regulation of gene means what? Gene is nothing but segment of DNA. From the gene, how protein is produced and how that is involving in some reactions or the processes, or how this particular gene is able to control some characters in the inheritance. What we are saying, from the parent to the offspring, the characters are moving. That's what we are saying, right? Characters are passing on from parent to offspring. Then how characters are passing? Originally, it is the DNA, it is the genes that are passing from parent to offspring. If genes are passing from parent to offspring, automatically characters are also passing. Then you need to understand that how gene is able to control the characters. Why you are observing different behaviors. All these things are interrelated with this particular concept. That is the reason why first we are studying the basic molecular mechanism. Once we understand molecular mechanism, then we can easily go for principles of inheritance and variation chapter, and that time you can very easily you can understand all the concepts. Is it clear? So that's why we are studying this one. And not only this, at the end of the chapter they have given two more concepts. One is regarding human genome project. So we are the human, and in each and every cell we are having the DNA, and what is the sequence of it? So scientists have started sequencing of this, and that particular ambitious project is called as human genome project. And after that, applications also they have given, and as part of it they have also given about DNA fingerprinting. So all these things we will study, and with that this chapter will be completed. This is what the basic introduction they have mentioned in the NCERT. So all these things, structure of DNA, DNA packaging, DNA replication, transcription, genetic code, translation, lack of run. This is about the regulation of gene expression. How exactly gene will undergo start and stop, that means on and off, that we will understand with the help of lac operon. And then human genome project, DNA fingerprinting. If we understand all these things, this chapter will be completed. That's it. So this is what they are saying. And now let us directly go with this NCERT content. Please remember, already in the introductory lecture itself I already told you, first we will go with the concept, and immediately after that we will read the NCERT lines, so that you can easily understand the NCERT information also. Generally what happens is, for the explanatory purpose we will use some, we may not use exactly as it is words of NCERT. We will give some other words so that you can easily understand the concept. But ultimately for the examination, you need to understand NCERT words also. So that is the reason why we will read NCERT also here itself, so that it will be easy for you. And now we are starting with the molecular basis of inheritance. Already I told you that the basic unit introduction and regarding scientists. We are not needing in this particular lecture. So as and when required, then we will go through that particular content. So now we are starting with the chapter. Chapter name is, or the title is, molecular basis of inheritance. For the inheritance of characters at the molecular level, what is responsible? That we need to study in this particular chapter, and how exactly they are working. Everything with molecular mechanisms we will study. Clear? Let us quickly go through this. In the previous chapter, what is the previous chapter as per NCERT? Principles of inheritance and variation. Okay, anyhow we have not completed that. First we are starting this one, so don't worry. In the previous chapter, you have learned the inheritance patterns and the genetic basis of such patterns. So actually in the principles of inheritance, what we will say, characters are passing from one generation to next generation, that is called inheritance, and for that inheritance genes are responsible. So genetic basis, genes are the basis, genes are responsible for transmission of characters, movement of characters from one generation to next generation. That's what we understood. At the time of Mendel, but who is called as father of genetics? Mendel. But actually, do actually Mendel know about these things? He don't know anything about the genes. He don't know anything about the genes. Simply he proposed that some factors will control the characters, that's it. So at the time of Mendel, the nature of those factors are nothing but genes. Nowadays we are calling them as genes or alleles. At the time of Mendel, what are these factors that are regulating the pattern of inheritance was not clear. Mendel don't know that what actually controlling the inheritance of character. He don't know anything. So over the next 100 years, so he proposed some things and then he died, and from the year of pronunciation of those particular Mendel's laws, from that approximately it took almost 100 years to know exact structure of that particular genetic material. Is it clear? That's what they are saying. Over the next 100 years, it took almost 100 years, 1 century, almost it took 1 century to exactly understand what is the genetic material and what is that structure. Understood or not? With structure also to know that. So in this way, over the next 100 years, the nature of putative genetic material. What is this putative word? Here I gave the meaning. So what is this putative? Supposed or considered. Please remember, at that time they don't know what is the genetic material. So they are considering something as a genetic material. That putative genetic material was investigated. So what is the genetic material? They have started investigating. So they have started experiments. All the experiments we will study in this chapter itself. Okay. So how scientists proved that DNA is the genetic material, everything we will study in this particular chapter. So they have started investigating, culminating in the realization that DNA means deoxyribonucleic acid, is the genetic material at least for the majority of organisms. What is this culmination means? It is the climax. So the scientists have predicted, they have started predicting that so and so may be the genetic material, and then they have started observing them. So for that they performed many experiments, and finally they got the climax. At the climax, what they have realized? They have realized that DNA, that is deoxyribonucleic acid, is the genetic material for most or majority of organisms. Majority. What will come under this majority? All 5 kingdoms and some viruses. Already we have discussed this in previous lecture. That's why in the previous lecture itself we have studied all these things. Next one. In the class 11th, you have learned that nucleic acids are polymers of nucleotides, whether it is DNA, RNA, whatever it is. Both of these are called as nucleic acids. Nucleic acids are polymers of nucleotides. That means many nucleotides will attach to each other and they will form a long polymer called a nucleic acid. That's what they have given here. Very simple information. So whatever is our regular class, that class content you need to interlink with NCERT. Then only your preparation will be the best. Okay. Now let us continue with the next paragraph. Deoxyribonucleic acid, that is DNA, and ribonucleic acid, RNA, are the 2 types of nucleic acids. Only 2 types of nucleic acids are found in living systems. Any living organism you just take. If these are present, only these 2 are present. That's it. Any living organism, only 2 types of nucleic acids are present. What are those? DNA and RNA. Next one. DNA acts as the genetic material in most of the organisms. DNA acts as genetic material in most of the organisms, majority of the organisms. What is this most or majority? It includes all 5 kingdoms and some viruses. Clear. Next one. RNA, though it also acts as genetic material. Can RNA act as genetic material? Yes. Not in all viruses, but in some viruses it is acting as genetic material. So in some viruses, it also can act as genetic material. What is the meaning of this also? So wherever DNA is not there, please remember this. Already we have discussed about this. If a particular cell is having both DNA and RNA, then who will act as genetic material? Only DNA. RNA cannot act as genetic material. But in case of some viruses, DNA is not present. DNA is not at all present. In that case, yes, RNA can act as genetic material. That's what the meaning is. So in this way, in those cases, RNA can act as genetic material. So overall, how many genetic materials are possible? Two genetic materials are possible. So actually, in the next coming lectures, we will study about this. In the NCERT, they have given approximately 1 page information, and there they are actually comparing between DNA and RNA. So both of them are genetic materials. Both of them are genetic materials. But why DNA is genetic material in majority of the organisms? Reasons, everything they have given in the next coming lecture. Slowly we will study. This is just introduction only. So let us complete it here like this only. So RNA can also act as genetic material, where in some viruses, material in some viruses. Observe this carefully, very clearly they gave some viruses, and that too mostly it is functioning as messenger. So acting as genetic material is one function, second one, and it is functioning as messenger. Yes or no? It is having messenger function. So mRNA, that's what they are talking. So if you want to underline messenger and write mRNA. Next one, RNA has additional roles as well. Not only this genetic material and not only messenger. It is having some extra other functions also. What are those? It functions as adapter. Underline adapter. What is that adapter? Connector. tRNA. So underline adapter and write tRNA. Next one, structural. Structural. What is this structural RNA? rRNA, ribosomes forming RNA. So underline structural and write rRNA. And in some cases, it acts as a catalytic molecule. Underline the word catalytic and write act as an enzyme. Catalyst means what? Enzyme. So now RNA acting as an enzyme is ribozyme. So underline catalytic, write ribozyme. Write enzyme hyphen ribozyme. RNA acting as an enzyme is called ribozyme. Already we have studied examples. Please remember, I already told you. So in this first page, this is the first page of MBI, right? Molecular basis of inheritance. And here you are getting some empty space. Here itself, please write all the 3 examples related to ribozyme. Whatever we have discussed, please write in textbook. Many times we will refer all these examples. Okay. So please remember this. Next, come back to this. So it is a catalyst. Next one, in class 11th, you have already learned the structures of nucleotides and the way these monomer units are linked to form nucleic acid polymers. Already we have studied this. Nucleotide structures already we have studied in biomolecules, and how nucleotides are attached to each other and how they are forming nucleic acid. Everything with detail already we have studied in class 11th biomolecules. But once again they gave in this chapter also. So again you need to study this. But generally what happens is, in the biomolecules of class 11th, we will not study with that much of details. There in simple we will try to complete that. But in this particular molecular basis of inheritance chapter, we need to study each and every point with structure also. With structure also. Why we need with structure? Because there are numerical questions that are possible. So in the examination, they are asking some numerical questions. In that numerical question, they are asking the calculation of the bonds. Yes or no? And if they are asking about the bonds, you need to definitely know about the structure. If you don't know the structure, unnecessarily you will face the problem. So we will study with the details. Even though they are saying that in class 11th we have studied, once again we will study each and everything with more details. Clear? Next one. So these are the structures. We know that nucleotides are the monomers and they are attached to each other so that they are forming the long polymers called nucleic acids. Clear? Next one. In this chapter of molecular basis of inheritance, we are going to discuss the structure of DNA. What we will study? Structure of DNA we will study. Its replication. It means DNA replication. How one DNA will make another DNA. That process also everything we will study. Even experiments are also there. That's why this is a very big chapter. Please remember this. You will get more than 50 lectures minimum. Okay, I am telling you in advance. So that much big. And not only that, the weightage of this chapter is very high. So one chapter of MBI, molecular basis of inheritance, is almost equal to entire human physiology. Please understand this. One chapter of MBI is almost equal to entire human physiology of class 11th. Now you can understand what is the weightage of this chapter. So obviously you can expect that this is the most biggest chapter in for this NEET UG course. Yes or no? So that's why Please be careful, and this is a very simple chapter because it is completely concept oriented. You don't need to by heart anything. Everything is completely concept oriented. Okay, next one. So DNA replication we will study, and next one, the process of making RNA from the DNA. Making RNA from DNA. Now observe this very carefully. So making RNA from the DNA, what is this process called as? Transcription. That means that particular transcription process also we will study. That's what they are saying. Next one, the genetic code also we will see, but here they have given function. What the genetic code will do? The genetic code that determines the sequence of amino acid in a protein. So now you just observe this particular sentence carefully. We are saying that from the mRNA protein is produced. Protein means it is polymer of amino acids. Many amino acids are there. But please remember, a particular protein always will have a particular sequence of amino acids. And now the point is how that sequence is decided. Do you have to understand this? How this sequence of amino acid is decided? That is based on genetic code. Genetic code will decide what is the sequence of amino acids. That's what the point they are saying. So anyhow, just before translation, that means just before protein synthesis, in the regular content we will cover genetic code, then we will go with the translation process. Everything we will study with the details. Don't worry. Okay, next one. Not only this, we will also study about the process of protein synthesis. What is the process of protein synthesis? It is nothing but translation. So that also we will study. And the elementary basis of their regulation, gene regulation, with the help of Lac-Operon we have written. No? So with the help of Lac-Operon we will study that how genes will undergo regulation, how they will undergo switch on and switch off, so that how they are able to control protein synthesis and stopping the synthesis. Both of them, how they are able to control, that thing also we will study. Next one, the determination of complete nucleotide sequence of human genome. So scientists have got complete nucleotide sequence for all the 46 chromosomes of human. Already we have that sequence. So scientists have done that. During the last decade has set in a new era of genomics. Scientists have already, so they have got the complete nucleotide sequence of human genome, and because of that, genomics, and this is a new branch that has started. Okay, so here we are discussing about many other things. So all these things also we will study, and in the last section, the essentials of human genome sequencing and its consequences will also be discussed. Finally, at the end of the chapter, we will study about what are the applications of this, and as part of that we will study DNA fingerprinting also. With this, the chapter will be completed. In this way, they are saying that what are the things that we are studying in this chapter. Over. Now what we need to study? Now let us begin our discussion by first understanding the structure of the most interesting molecule in the living system, that is DNA. So till now we have studied about what is the content we will study in this chapter, and now we are starting with the regular content. So in this content, first we are starting with the structure of DNA. This is what they are saying. So definitely, to know the structure of DNA, first you need to know the structure of a nucleotide. So that's what we are starting now. Okay, next one. In subsequent sections, means in the next coming sections, we will understand that why it is the most abundant genetic material and what is its relationship with DNA, RNA. So first we will study about the structure of DNA. Once we are completing that, we will study about what is the comparison with RNA and why most of the organisms are having DNA as genetic material. Reasons also we need, right? So all these things slowly we will study in the next coming sections. This is what they are saying. With this paragraph is completed, and now we are going with some explanation. And please remember, this explanation will take so much of time. So maybe in 1 or 2 lectures, I will try to complete this complete explanation. Once this explanation is completed, then only we will go with NCERT reading. Until then, I will not do the NCERT reading. Okay, it may take 1 or 2 lectures more. So then we will go with that particular content. Okay, clear. And now we are going with the nucleotide. In this lecture, first I will give some basic introduction itself. That's it. And original structures we will start from next lecture onwards. And here simply I will make one particular flow chart, and with that this particular lecture will be completed. Is it clear? And now please remember, now we need to understand the structure of nucleic acid. Please remember, this basic information is common for both DNA and RNA. The way the DNA and RNA are formed, mechanisms are almost same. Only small differences are there, that's it. If you understand those differences, that is enough. Understood or not? So now we are going with this particular structure of DNA and the structure of RNA. So that's why instead of saying about DNA or RNA separately, simply you can say the structure of nucleic acid. Structure of nucleic acid. And now you need to understand the heading of NCERT. This is what the beauty of NCERT. Please understand this. In the heading of the NCERT, they have not given structure of DNA, they have not given structure of RNA, they have not given structure of nucleic acid also, they have not given. Then what they have given? They have given the structure of polynucleotide. Please understand the words. Polynucleotide. Please understand this. Understood or not? Generally, we are expecting DNA and RNA names. Okay, they have not given, fine. At least we are expecting the name of nucleic acid. That also they have not given. Instead of giving nucleic acid, they have given a word called as a polynucleotide. Structure of polynucleotide. Now, what is the meaning of this one? Poly means many. Many nucleotides are attached to each other. This is one nucleotide. And now to this, another nucleotide, another nucleotide, another nucleotide, another nucleotide, like this. Now, in this way, if many are attached, now this is called polynucleotide. What is polynucleotide? It is nothing but nucleic acid. It is nothing but nucleic acid. If these nucleotides are related to DNA, now this molecule will be DNA. If these nucleotides are related to RNA, this molecule will be RNA. That means in the NCERT itself, in general, they are giving information for both. Because this basic information is common for both DNA and RNA. So what is the small difference? That we will study separately. But remaining 99.99% information is same for both DNA and RNA. So that's why here itself we are studying for both DNA and RNA combinedly. Is it clear? So anyhow, if you want to understand the structure of DNA or RNA, the basic thing we need to understand is structure of nucleotide. Each monomer, each single unit is called as nucleotide. Spelling is very important. TIDE, tide. So that's why first we need to understand nucleotide very clearly. Once you understand the nucleotide, that's it. 80% information is completed. And remaining 20% will include linking bonds. What are the bonds that are formed? And between what type of molecules, what type of bond is present. Those points we need to observe separately. That's it. So that's why this is the most important. So now we are going with the nucleotide. Nucleotide. Now we will make a small tree diagram. So now let us observe each and every point very clearly, and with that we will conclude this particular lecture. Let us see. Nucleotide. Tide has three components. Three. Understand this. So already you have studied about biomolecules. So you know about two names. One is nucleotide, TID, tide, and other one is nucleoside, SID, side. Yes or no? So nucleotide and nucleoside. Nucleoside. Please remember, spellings are almost close, but here T is there, here S is there. But there is a huge difference between these two. Please understand this. And here, which one is the monomer? What is the basic repeating unit? Is this nucleotide or nucleoside? What is the correct answer? It is the nucleotide. It is not nucleoside. So this nucleoside, maybe in the next lecture we will study. Okay, here now we don't want to study. So it is the nucleotide. And this nucleotide has how many components? Three components. Three components. First one, second one, and the third one. So what is this first one component? Please remember them in the same sequence only, so that when you are writing the structures, it will become easy. The three you can write in any way. Any way you can write, no problem. Ultimately, you will write the same three only. But what is the sequence in which I am writing? Remember that. So that in the next lecture, when we are studying the structures and when we are linking these molecules, at that time it will become easy for you. Clear? Next one. First one. What is the first one? Nitrogenous base. First one is nitrogenous. Generally, we will write simply N. Okay, nitrogenous base. Generally, when we are writing, we will write N base. N base means nitrogenous base. Instead of writing a long one, we are writing like this. Next one. Next one is a sugar residue. Next one is a sugar. Next one is phosphate group. Phosphate group. In this way, nucleotide has three components. What are the three components? Nitrogenous base, sugar, and then phosphate. These are the three things. Now, overall, if you see the DNA and RNA, please remember this information we are writing for both DNA and RNA combinedly. So, how many types of nitrogenous bases are we observing? Based on the structure of these nitrogenous bases, please understand this. Based on the basic structure of these nitrogenous bases, two types we are writing here. One is purines, and the other one is pyrimidines. Pyrimidines. See, you can pronounce this as a pyrimidines or you can pronounce this as a pyrimidines. Anyhow, you can pronounce this, but I am pronouncing this as a pyrimidines. Okay. So, nitrogenous bases we are classifying into two types. Nitrogenous bases are classified into two types, purines and pyrimidines. And again in the purines, if you are observing this, based on the small structural differences, again these are two types. One is called as adenine and other one is called as guanine. One is adenine and other one is guanine. For the adenine, the standard abbreviation is A. First letter we are using. For all these things we are using first letter. For adenine, A. For the guanine, we are indicating with G. That's it. So these are the two types of purines that we are observing in the nucleic acid. Please remember, many students are thinking that in the cell if they are asking, more than this are possible. But our question is, how many types of purines are present in nucleic acids, nucleotides, nucleic acids, like that we are talking. Understood or not? But other forms of purines may be present in the cell. So that's why read the question very carefully. Then only you will get the proper clarity regarding the question. Once you understand the question clearly, then only you can go for the correct answer. Please understand this. We are studying about these types only present within the nucleic acids. There are some other molecules. Clearly I am telling you this. As in when required, at that time I will tell you. There are some other types of molecules. They may be purines, they may be pyrimidines, but they may not be present in the nucleic acid. And those molecules we are not studying here. Please understand this. We are studying only those purines, those pyrimidines which are present in the nucleic acids. This is the point that you need to understand. Clear? So purines two types, adenine and guanine. Only these are present. Next one. But pyrimidines, overall for DNA and RNA combinedly, when we see three types of pyrimidines are present. What is the first one? Cytosine. First one is cytosine and it is indicated with capital C because first letter is C. Okay. Next one. Next one is uracil. And it is indicated with U. And after this, the third one is thymine. Okay. Thymine. Now this spelling is important. I will come to this particular one later on. First we will see the spelling important. What is the spelling of thymine? I will give the two options. Observe this carefully. I will give you two options. First one is THYMINE. THYMINE. It is called thymine. And this is THIAMINE. 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 Whereas THYMINE, that is thymine. Now tell me. Please remember, these two are completely different. Now you need to tell me which one is used here. What is the spelling of this pyrimidine? Spellings are very important. Many to see. Sometimes spellings will not make any matter, but sometimes spellings are important. Based on these spellings, English grammar usage, sentence reading, for all these things, I made a separate video. I already told you this. So please go through those introductory lectures, introductory general information related videos. Complete that first and come back to this. Okay. And in this case, spelling matters. Because these two spellings will give two different meanings. Please understand this. First, THIAMINE, somewhere separately you just write this. Okay. Anyhow, in the separate notes, whatever we are uploading, in that I will mention, but if you want to write, you just write. THIAMINE, thiamine. What is this? This is a vitamin. Anyhow, in your syllabus of NEET examination, so regarding the vitamins, it was not mentioned. So I don't want to go into more details. Okay. So actually THIAMINE is a vitamin. You can call it as a vitamin or vitamin. Okay. So this is a vitamin. But what about this one? This is the nitrogenous base. This is the nitrogenous base. THYMINA thymine is the nitrogenous base that we need to write here. So please observe this. Spelling is important, and below this Y you please underline so that you can remember this. Spelling is important. THYMINE. So in this way, pyrimidines 3 types are present, and this thymine is indicated with capital T. Understood or not? So nitrogenous bases are 2 types, broadly classified into 2 types. If those which are having purine ring, are called as purines. Those which are having pyrimidine ring are called as pyrimidines. That's it. Now within the purine ring again, based on the substitutions again, 2 types are there, adenine, guanine. Now here all the 3 are having basic pyrimidine ring, but based on the differences in the substituted molecules, now again there are 3 types: cytosine, uracil and thymine. Spelling of thymine is THYMINA thymine. That's it. So this is about the nitrogenous bases. Now we are going with the sugar. Now here, so since we are combinedly studying for DNA and RNA, overall how many types of sugars? 2 types of sugars. I already told you this. Ribose sugar containing molecule, ribonucleic acid. Deoxyribose sugar containing, so DNA. Yes or no? So same here. So some types of nucleic acids are having ribose sugar and they are called as ribonucleic acid, RNA. Whereas some other types of nucleic acids are having deoxyribose sugar, and they are called as DNA. And these were 2 types. Structurally almost similar, small difference is there. In the next lecture we will study about the structures. Okay, and then phosphate group. You can call this phosphate as orthophosphoric acid. Orthophosphoric acid, ric acid. Please remember here. So once in the NEET examination, they have not asked in the biology, but they have asked in the chemistry. In the chemistry also biomolecules is there. So that's why this point is very, very important. So there what they did is, they said that DNA has phosphorus acid. This is what the statement they give. DNA has a phosphorus acid in its structure. True or false? That is false. That is false. Phosphorus acid is different. Phosphoric acid is different. And what actually the DNA is having? DNA means nucleotides. Nucleotide means phosphoric acid. You can also call this as orthophosphoric acid, or simply you can also call it as phosphate group. Okay. Anyhow, more details with structures, everything we will study in the next lecture. This is what the basic information. So now let us conclude this. Nucleic acids are 2 types, DNA and RNA. They are the polymers of nucleotides. So each monomer is called as nucleotide. So now first we are studying the structure of each monomer, that is called nucleotide. Nucleotide has 3 components. First one is nitrogenous base, second one sugar, third one phosphate. Please remember in the same sequence so that in the next coming lectures it will become very easy for you. Nitrogenous base is 2 types, purines and pyrimidines. Purines are again 2 types, adenine, guanine. Adenine is indicated with A, guanine is indicated with G. And pyrimidines are 3 types, cytosine, C, uracil, U, and thymine, T. These are the 3 types of pyrimidines that are present in the nucleic acids. Next one, sugar, 2 types, ribose, deoxyribose. Ribose is seen in RNA, deoxyribose is seen in DNA. Next, phosphate group. It is also called as phosphoric acid. It is also called as orthophosphoric acid. Anything you can say this. Okay. So this is what. And regarding these things, for each and everything with the structures, we need to study in the next lecture. Okay. If possible, in the next lecture, we will try to complete these structures. Okay. Or else as far as maximum as possible, so we can complete these structures. Once we complete these individual structures, then we can go for the next remaining content. Okay. So with this particular lecture we are stopping. Okay. Thank you so much, my dear friends. We will meet in the next lecture. Thank you.